Meet Sarah and Ali. They're both prescribed the same dose of tacrolimus, a drug used to prevent organ rejection after a transplant. But something surprising happens. The level of tacrolimus in Sarah's body stays stable, and the drug works as expected. Ali's level, however, spikes dangerously high, putting him at risk of serious side effects. Why the difference? The answer could be written in their genes. In this video, we're exploring how genetics influences the CYP3A enzyme system, and how this variation can affect drug metabolism, safety, and efficacy. The influence of genetics on drug behavior is known as pharmacogenomics. Pharmacogenomics is the study of how a person's genetic makeup affects their response to drugs. It combines pharmacology and genomics to help explain why people metabolize medications differently and how we can use that information to make prescribing safer and more tailored for the individual. Let's start with a key concept in pharmacogenomics, the single nucleotide polymorphism, or SNP, pronounced SNP. A SNP is a change in just one letter of the DNA code, A, T, C, or G. These variations can happen anywhere in the genome, and they're common, found in at least 1% of the population. Some SNPs occur in the CYP3A4 gene, which provides instructions for making the CYP3A4 enzyme, the most active member of the CYP3A family. SNPs in CYP3A4 can lead to genetic polymorphisms, inherited differences in how much of the enzyme is made or how well it works. Most of these polymorphisms are small, but even subtle changes can impact how a drug is metabolized. For example, one person's version of CYP3A4 might metabolize a medicine quickly, reducing its effectiveness. Another person's enzyme might work more slowly, leading to higher drug levels and a greater risk of side effects. These differences help explain why the same dose doesn't always work the same way for everyone. Some well-known CYP3A4 polymorphisms, like CYP3A4*22, are associated with reduced enzyme activity. Carriers of this variant may need lower doses of certain medications to avoid toxicity. It's not just CYP3A4. Another gene, CYP3A5, is also involved. Some people carry a functional version of CYP3A5 and metabolize certain drugs faster. Others lack functional CYP3A5 altogether, changing their overall metabolic profile. Tacrolimus is a good example. It's metabolized by both CYP3A4 and CYP3A5. Patients with functional CYP3A5 often need higher doses to achieve the same effect. Without it, lower doses are usually safer. Pharmacogenomic testing can help identify these differences before prescribing. In the future, we may routinely tailor doses and drug choices based on a patient's genetic profile, reducing trial and error and making treatment safer from the start. Understanding how genetics shapes CYP3A activity is one more way we can personalize care and improve prescribing decisions.